Welcome to this technical guide on using HALO-96 PRS and Luminesce-96 PRS and how to establish a cell potency reference standard. I'm Ivan Rich, CEO of Hematopoietic, and I'll be accompanying you through this presentation. There are three things that you need to measure potency. The first is a validated readout system that measures biological function. The second is the ability to specifically measure the stem cell active constituents or components in a dose-dependent manner. And finally, you'll need a reference standard of the same material as the sample. That's what this video is all about. Establishing a cell reference standard for a potency assay is often considered the most challenging aspect of the assay. In the United States, reference materials are a requirement for measurement of potency, since these are embedded in the Code of Federal Regulations. But they're also a requirement of other regulatory agencies. A reference standard is needed because it allows you to compare the identity and biological activity of the active components that will ensure product stability and consistency between lots or batches during the manufacturing process. It allows evaluation and/or correlation with a clinical response. It can help predict and avoid product failure or toxicity due to improper potency. It helps predict whether the product can be released for use. And finally, it helps predict the dose required to ensure the product will produce the intended effect or response. Now, there are problems establishing a reference standard for a cell therapeutic product. In contrast to traditional drugs, where the reference standard can be obtained in relatively large quantities and purchased from the US or European pharmacopoeia, or even from the World Health Organization, for example, there is insufficient material from a cell therapeutic product to produce a global reference standard for a specific product. There are no standard cell preparations because there is little consensus on how to prepare cells in a standardized and consistent manner, even for the same cell product. In many cases, standardized and validated assays are unavailable that would allow direct comparison of results to help prepare standardized cell preparations for use in potency assays. At the present time, there is probably only one avenue open to establish a reference standard for a specific cell therapeutic product, and that is to establish the reference standard in-house. Reference standards are established as primary, secondary, and even tertiary reference standards. That means that a primary batch of material or cells is processed, aliquoted, and stored. And for cells, they would be cryopreserved. And the identity and biological activity of the active components measured using a standardized and validated assay. This would establish the primary reference standard. A secondary batch of the same material or cells would be processed, the identity and biological activity of the active components compared to the primary reference standard used using the same assay. This would be the secondary reference standard. A similar process would be used to establish the tertiary reference standard. Now, establishing a new reference standard of the same material or cells is always performed against its predecessor reference standard. The last reference standard established is for everyday use. Since the same standardized and validated assay would be used for the reference standard and the samples to be tested, the potency can be measured directly. In this way, we at Hematopoietic have established our own in-house reference standards for bone marrow, cord blood, mobilized peripheral blood, and mesenchymal stem cells. Now, there are requirements for using a reference standard to measure potency. The reference standard must be of the same material and contain the same active components as the sample being measured. That is, if a sample is cord blood, the reference standard must also be cord blood. If the sample is a CD34 preparation from bone marrow, the reference standard must also be a CD34 preparation from bone marrow. And if mesenchymal stem cells are derived from adipose tissue, for example, then the mesenchymal stem cell reference standard will also have to be derived from adipose tissue. Whenever a product is to be used, a potency assay employing a reference standard must be performed together with the sample before release. Failure to incorporate the reference standard means that the potency ratio cannot be ascertained and the criteria used to release the product cannot be adhered to. The assay used to measure both reference standard and sample must be the same. The assay parameters must allow establishment of acceptance-rejection limits for both the assay itself and the cells it measures. Now let's turn to the two assays that are used to establish reference standards: HALO-96 PRS and Luminesque 96 PRS. These assay kits are used to establish in-house reference standards for hematopoietic stem cells from any source using the HALO-96 PRS and mesenchymal stem cells from any source using the Luminesque 96 PRS. The assay kits are available in 1 or 2 plate sizes. The assay kits include a cryopreserved vial of cells of the same source as the in-house reference standard to be established. Both assays incorporate the same standardized and validated ATP bioluminescence signal detection readout. Both assays measure the proliferation potential of the stem cell populations. Proliferation potential for proliferating populations, such as stem cells, is a measure of their primitiveness. The more primitive the cells, the greater the proliferation potential. It also follows that the more primitive the cells, the greater its potency. Therefore, potency is also a measure of proliferation potential. Proliferation potential can only be measured using a cell proliferation assay in which the slope of the cell dose response is determined. And the potency ratio is the ratio between the slope of the sample compared to the slope of the reference standard. The process of establishing reference standards using HALO-96 PRS, and in fact Luminesque 96 PRS, is shown in this flow diagram. First, obtain the primary hematopoietic stem cell source, or the mesenchymal stem cell source. Process the cells to a mononuclear cell fraction to ensure that the red blood cell concentration is about 10% or less. The mononuclear cell fraction is important because it does not contain dead or contaminating cells that can dilute the active stem cell components. The low red blood cell concentration is important because red blood cells interfere with the ATP bioluminescence readout and cause false positive results. Once the cells have been processed, aliquot the cells and cryopreserve them. After a certain period of time, remove an aliquot of the cells and thaw them together with the reference cells provided in the HALO-96 PRS kit. Perform a minimum of a 3-point cell dose response curve for 2 stem cell populations from both thawed cell aliquots using the HALO-96 PRS assay kit. This will only be 1 cell population for mesenchymal stem cells. For hematopoietic cell therapy products, 2 stem cell populations will be measured. The first is a primitive lymphohematopoietic stem cell population designated HPP-SP that can give rise to both lymphopoietic and hematopoietic cells. And the second is a primitive hematopoietic stem cell population, CFC-GEMM, that gives rise to the hematopoietic lineages only. The slope of the linear regression for each stem cell population measured provides the proliferation potential and therefore the potency of the stem cell population. Compare the proliferation potential of the established primary reference standard to that of the PRS reference cells. After the first primary reference standard has been established, prepare a second batch of reference standard cells in exactly the same manner. The HALO-96 PRS assay kit usually only needs to be used once, because in establishing the second reference standard, an aliquot of the primary reference standard, or the in-house primary reference standard, is now used to establish that secondary reference standard using the HALO-96 PQR assay. Now, after thawing, perform a cell count and viability. The viability after thawing should be equal to or greater than 85%. Based on the mononuclear cell count, perform a serial dilution to produce 3 working cell concentrations from the PRS cell suspension and the same cell concentration from the new primary reference standard. The working cell concentrations will be 750,000 cells, 500,000 cells, and 250,000 cells per milliliter. Dispense 0.9 milliliters of the HPP SP master mix into each of 6 tubes, 3 for the PRS assay and 3 for the new primary reference standard assay. Do the same for the CFC GEMM master mix. Now dispense 0.1 milliliters of the PRS cell dilutions into the respective tubes. This dilutes the working cell concentrations tenfold. The cell dilutions for the new primary reference standard can then be dispensed. After mixing the contents of each tube, 8 replicate wells are dispensed into columns across the plate as shown in the diagram. This further dilutes the cell doses to the required final cell concentrations. 8 replicates are used because statistics are very important in this assay, and if outliers have to be removed, sufficient wells should remain to provide clear-cut results. The cells are incubated for 5, 6, or 7 days depending on the work schedule. There is about a 3-fold increase in proliferation from day 5 to day 7, since the stem cells are proliferating exponentially at this point. Calibration and standardization is a requirement for a potency assay. All HALO assays incorporate an ATP bioluminescence readout that includes ATP standards and controls. Please see the technical guide videos on how to perform an ATP standard curve and controls and how to process the sample to measure cell proliferation. Now let's take a look at some of the expected results for hematopoietic cells. The diagram shows the ATP results for 5 potential cord blood stem cell reference standard preparations enumerated on day 7 of culture for the primitive hematopoietic stem cell in the upper graph and the primitive lymphohematopoietic stem cells in the lower graph. Since a potency assay cannot be measured as a single point and must be performed as a cell dose response, the linear regressions of each cell dose response at 2500, 5000, and 7500 cells per well is shown. The black cell dose response represents our primary cord blood reference standard. Since hematopoietic stem cells are the only cells that can initiate an engraftment process, the stem cells are the active components. The potency of a stem cell is measured by its proliferation potential, which in turn is measured by the slope of the cell dose response. Potency is measured by the potency ratio, which is calculated by the slope of the sample divided by the slope of the reference standard. For CFC GEMM, there are 2 samples with a greater potency ratio, while for HPP SP there are 3 samples, one of which is similar to the reference standard. Any of these samples could be used as a secondary reference standard, but in general, a sample that is similar or greater than the present reference standard would be used. Now let's take a look at some of the expected results for mesenchymal stem cells using Luminesque 96 PRS. Establishing a mesenchymal reference standard using the Luminesque 96 PRS assay is performed in exactly the same way as for hematopoietic cells, except that there's usually only one population to be measured. The upper graph shows the difference in cell dose response slopes for bone marrow derived mesenchymal stem cells as a function of cell passage. The steeper the slope, the greater the mesenchymal stem cell proliferation potential and the greater the potency. The passage of mesenchymal stem cells used to establish a reference standard is an important factor to take into account and may have to be accompanied by a secondary assay such as flow cytometric analysis that can be multiplexed with Luminesque 96 PRS. The lower graph shows a bone marrow derived mesenchymal stem cell reference standard in black from passage 2 and compared to other mesenchymal stem cell preparations also at passage 2. The potency ratio for all mesenchymal stem cell samples has been calculated. Both MSC number 2 and 3 could be used as mesenchymal stem cell reference standards. So now you have the information that allows you to establish reference standards in your own lab. The final task is to actually measure the potency of samples and understand what the information is telling you. That's discussed in the last technical guide video of this series. Thank you.